Okay, thank you very much, Olivier. As you know, I am quite enthusiastic, but I think it was one of the most beautiful stories that I heard in my whole life. Really, congratulations. So, any questions, Martin?
I can also speak without microphone. You showed very nicely that there is a difference in the phenotype of your tumor depending on the expression level of the fusion protein. But do you know how the expression level is differentially regulated within the tumor?
So, no. It's a quick answer, no. It's a short answer. Other answer is, we know some, I spoke about hypoxia. So within the tumor, hypoxic areas lead to chronic hypoxia will lead to a decreased expression TWS1, so decreased activity of TWS1. But there are probably many other factors that contribute to differential activity. But you have to think that differential activity may be at the transcription level, the expression, at the protein stability level, or downstream. And it is known, for instance, that some downstream transcription factor, like YAPTAZ, TED, can counteract what EWS1 is doing. So may explain also a decreased expression of EWS1. The Wnt signaling pathway can also lead to a decreased expression of, not decreased expression, the expression remains constant, but decreased activity by competing downstream with EWS1. So this is a whole field to be explored. We probably have only a very few insights into this.
Yes. Otherwise, you won't hear me. Okay, so a great talk, as usual, I would say. Just a question on the tumor microenvironment in the Ewing sarcoma, because the tumor microenvironment, of course, you have hypoxia, so huge lot of blood vessels. But you have also is neurons and neurogenesis problem. Because the so in the Ewing cells you have so expression of NGF in all these factors, and now as you know the neural so interaction, the nervous interaction with the tumor cells starts to be so quite a focus of a lot of people in not only central nervous tumors but also breast. So that's called tumor types. So what's the situation of the Ewing sarcoma?
Yeah, so I didn't present this, but we did some single cell analysis of Ewing tumor, looking at the microenvironment. So this is immunology cold tumor. We only observe some macrophage with the phenotype of repressive macrophage for immune cells, very little T cells, very little B cells, no neurons, but this may be just linked to the way this is done, the landscape of cell analysis. But I think the pathologists may also say that in Ewing sarcoma, it's mostly a very homogeneous tumor, round cell tumors with very little other tumor types in the tumor. I'm speaking about the control of the bus, of the two buses.
So okay, just another small one. The EWS
EF1.
right? So the downstream genes, you said there's a lot of downstream genes regulated a lot. So you have I don't know, hundreds and stuff like this.
Yeah.
So did you do a hierarchical classification a little bit, which would be, let's say, the most important because I know, I read that you have also thrombospondin is a downstream. It's a downstream, and thrombospondin is pretty important microenvironment.
Yes, so I didn't speak about this, but we have number of genes involved in cell proliferation, like for instance, cyclin D1 and number of other genes. We have genes involved in neural differentiation, meaning that EWS5 by itself pushes also towards some kind of neural differentiation. And initially, I'm still speaking of those controlled pathologies, but Ewing sarcoma was described as a tumor of neural origin, neural crest. What we believe now is that it's not the neural crest derived tumors, but that EWS5, this has been shown by transfection experiment, induce the expression of some neural genes, which may lead to this kind of phenotype of neural phenotype. And many other genes.
And do you have?
And what is interesting, just to continue on this, is that when you look at different cell lines and you do the knockdown of EWS, and then you profile those cells by RNA-seq, you will see that those signatures are very divergent from one cell line to the other. And at the end, you only remain with a core common signature, which is quite small. One of the hypotheses for this will be that there is a lot of polymorphism in this kind of microsatellite. And from one tumor to the other, the distribution and the polymorphism of microsatellites may be different, leading to distinct target genes downstream.
And do you have EWS fly expression in the brain?
You don't have EWS5 anywhere, apart from the Ewing cell.
You only have in the Ewing cells, and it cannot be activated elsewhere.
No, because this is a translocation.
Okay.
So unless you have a translocation in the brain, but there is no really Ewing in the brain.
Thank you very much for this very related talk. As you showed that the microsatellite sequence are spread in subtypes in some chromosomes, and this may be a general mechanism of activation of new genes, not only for Ewing sarcoma, but for other tumors. Could you imagine a therapeutical approach to knock down this interaction between this transcription factor and microsatellite?
Well, this is something we are trying to do. The first idea was to use synthetic oligos, double-stranded, and transfect the Ewing cell. But unfortunately, and trying to compete with EWS5. But at least in our hands, this didn't work. Probably because we could not really get the oligo into the nucleus. So this is one of the ideas. One of my colleagues has an interesting idea, is to have a vector with toxic genes, suicide genes, controlled by microsatellites. And then we put this to an antivirus, something kind of virus, and inject this, saying that only Ewing cells will be able to activate this suicide gene and be killed. This is an interesting hypothesis. Elegant, could be elegant. And this is Thomas Gunvald, one of the former postdocs in the lab.
But then it's not so easy to give or to target cancer cells, or you have to talk with François.
Well,
the viral particles of the right cell.
Yeah.
Any question? Enfin, tu veux le micro?
Yes. It's okay. Is this translocation a one-step transformation process, or do your mesenchymal stem cells already have something like p53 alterations or something like that?
No, most of the Ewing cells, Ewing tumors, do not have anything else than EWS.
Well,
We did a lot of 100 whole genome sequencing.
for RB, you can understand, like, can you make basically a effect on RB
Yeah.
for P53?
And P53 is frequent in cell lines, but rare in patients. Only 5% something of the patients have P53 mutation. And they have a very poor prognosis. But P53 is clearly not essential for EWS flag.
It's like KDM5 or whatever.
No,
or it's
no.
BRD8, which was.
No, STAC2 I presented briefly is the most frequent guy. To be the tool that we do.
And for your model, you need co-mutation. You need to do co-mutation of CDKN2A, P53, STAC2, and
Yes.
doing or only a few
Initially,
actors.
not now. This was the initial experiment. Now, this is the work we did with Erika Brunet. Some of you imagine. And initially, we said we wanted to have the maximum of change on our side. So we use STAC2 inactivation and P53 mutation. But now they can only use EWS line translocation to transform those cells.
You very elegantly showed that the GWAS studies identified these SNPs and this also repeats. And so my question is, this is an association study, but
Yeah.
do you have insights about the link between this microsatellite and the generation of the translocation in Ewing sarcoma? Maybe is there something at the mechanistic point of view? Is there something possible that makes a link between the susceptibility or association with the GWAS and the mechanism of
This is
translocation?
an important question. When I began the GWAS, I suppose, or I hypothesize that the main loci that may show up will be chromosome 22. Something on this region that distinguish Ewing patient from the genetic. But we just observe nothing at the level of chromosome 22. So the idea that we have now is not that those polymorphisms facilitate the translocation, but facilitate the activity of the fusion gene through a polymorphism of macrophages.
And also one comment is very interesting. You showed about the tuning of the levels of activity of the fusion in cells determines the cell fate and so on. Do you believe that this is something common to many oncogenes? Because, for example, we have a collaboration with David Santamaria, the colleague of Marielle Barbacid, and we have similar observations for the RAS-MAP kinase pathway that is what David calls the sweet spot. So you need too little is not good, too much is also not good of an oncogene, as we discussed about oncogenic toxicity today also.
I think that in melanoma, it has been shown that you also have differential activity of MITF. And that this could be something very important for the biology of melanoma. So this is not the only example of this kind. So just meaning that this is not oncogene or not oncogene. There is all a range between the two, and you need probably this kind of plasticity for the.
Yeah, as we discussed, we were very naive, I think still a bit naive somehow, that we believe that putting an activity of an oncogene would transform the cell, but then it's a lot more complicated.
I'm always very intrigued by benign soft tissue tumors that have, at least for a number of them, translocation, that have fusion genes, but that are benign. And is it because you do not have this shift? Because I believe this shift is essential for you. If you do not have this shift, you may not have metastasis. So maybe something to explore.
Well, I think we are over. We can let Olivier brace a little bit because it was a very marathonian day for him. So once again, we thank you so much. Thank you very much. And thank you all for participating.